Okay, very good evening, class. Sorry for a bit delay. I was just trying to fix the things regarding this magnification. I hope my voice is clear, loud, and the screen is also good. There is sometimes problem that I had. Uh, no, I was just trying to fix the things to make it to 720p quality. So that's what took me some time. Um, okay, so we should not waste our time much. I hope everyone has joined. Things are on the road. Everything is well. So there was some confusion in the group about writing the assignment. Bache, it's like when we are done with one concept, like I want to join basics of your molecular biology with the cells. So you make something together with the cells and the basics of molecular biology and one assignment out of it. So the day I will tell you that okay, now you're good to go to make assignment. So on that day, you can start with it. Okay, nice. So I hope you're all doing well. Everything is fine at your end. Yesterday we were talking about DNA. We have to start about DNA actually. So we will focus mainly about your trans molecular biology. That what's the basic molecular biology includes, all includes about. So let's start with DNA. So as we have discussed, it's a nucleic acid that contains the genetic instructions into the development and functionings of the all living organisms and viruses. So that's what the DNA is made up of. That's what the DNA is all composed of, which at the end become a blueprint to construct the components of various cells, such as your proteins, RNA, and other molecules. So based on that, everything works out. So basically, we see that DNA, it's a two strands, double helix structures. Like here we can see this is double helix structure here, and sorry for the images, I need to update them just due to the lack of time. But here, but still we can see that there is a G, C, A and T. So there is a pairing of guanine with cytosine and adenine with thymine. So like this, they make all these pairings, and the radius is around 1 nanometer, and the total diameter is around 2 nanometer. And if you see from the including your major groove and minor groove, so together it's make the height of 3.5 nanometer of one loop. And here we can see some hydrogen bonds are happening between thymine and adenine, and it's mostly the hydrogen bonds that able to make them and gives them a double hydrogen bond. And also there's a triple hydrogen bond between G and C. Because of these hydrogen bonds that is happening between your purines and pyrimidine base pairs, we can have this helical structure. That's why the reason, just because of these bondings in the centers, the DNA is making this helical structure. Otherwise, it might be not possible. And beyond that, what you can see beyond these nucleotides here, thymine and adenine, guanine and cytosine, that there is some sugar, ribose sugar, deoxyribose sugar, and they are being connected with the help of your phosphate group. And this way, there is a side backbone chain is being made in this parallel, and on the other way. And in this side, on the left side, it's going from 5 prime end, because 5 carbon we take from here, and it's over at 3 prime carbon end. On the other side, it goes opposite from 3 prime end to the 5 prime carbon end. So this way, we have this anti-parallel structure of our DNA, which is a polymers of your nucleotides and also backbone made of a base of sugars and phosphate groups. So what is this is very common confusions. Mostly students get confused. Even they went for PhD. Even sometimes in our interview for the it's a funny thing. We were asked in our interview that can you give us a difference between nucleosides and nucleotides, because once you study too much in your life, but the interviewer, the person who is on the other side, always wanted to see that whether your basics are clear or not. So that's why this presentation will be really helpful for you, and the assignment that you will make might be forever important for you, many perspective. Because I have been through a lot of interviews also, I have been through a lot of academic life also, I have been through some industrial life also. Now I'm in pure academia based on teaching and these research papers. How this world works, basically it all works if your basics are clear. Once your basics are clear, things will be very easy for you to overnotch any aspect of your life. Yeah. So that's why I make my first or 2 3 days keep speaking same thing again and again, even though I know it's very basics from your class 12th. But I want to make your base strong so that we can make a army of our knowledge even more stronger. Yeah. So here we can see there's a nucleoside. Nucleoside is basically made up of your sugar and base pair. So you have sugar here and the base pair together, they will make a nucleoside. But into that same sugar base pair, if you attach your phosphate group, yeah, into the sugar nitrogenous base group, if you add phosphate group, it's just become nucleotide. So that's the major difference between your side and tide here. Sugar base is nucleoside, phosphate plus sugar plus base is your nucleotide. And what are the various nitrogen base pairs we are having actually? So it's mainly your purines and pyrimidines. Sorry for this. So here we can see the purines includes your basically 5 and 6 membered heterocyclic compounds, whereas the pyrimidines includes mainly your 6 membered rings. So in purines, mainly it's containing Ag. You can learn it. Purines means pure, and Ag make some rhyming with it. Ag pure, something like that. So you can remember that Ag is always purines, or something with Ct pyrimidines, like pyrimidines are going pyramids, or something with Ct. You can make a whistle at the pyramids. So you can combine them. You can try your own way to remember these stuffs. Sometimes for some it's difficult. So yeah, so adenine includes your 5 to 6 base pairs. So you have a 6 membered ring here with the nitrogenous groups, amine groups present, and also this amine group at the present on the 6th carbon here. And this is guanine with the oxygen group here present, and also one amine group present. And whereas the cytosine and thymines are present in the DNA, but in the RNA it's uracil replace your thymine, and instead and the cytosine and uracil, sorry, thymine and the uracil, the difference between a thymine and uracil is basically the methyl group. The methyl group is absent, and that makes it to the uracil. So fifth pyrimidine, as I said, uracil usually takes place thymine in the RNA, and just differs from the methyl group there. And basically all the pairing are happening between your purines and pyrimidines together. So A will pair with the T and C will pair with the G. So purines and pyrimidines pairing with each other. And how they are pairing? So if we say adenine and guanine, they will be making this double bond here, whereas guanine and cytosine might be making this triple bond. Yeah. So and how the sugar backbone, that change that we see in the DNA and RNA, how is that differs? Why RNA is ribose nucleic acid and why DNA is deoxyribonucleic acids? It's just because of the carbon atom at the second carbon atom in the ring. It might not have this hydroxyl group. So in the RNA, the hydroxyl group is present at the second part, and whereas in the DNA, it's absent. And the rest is the same ribose sugars looks like. So there's also major groove and minor groove. So there's a distance between each base pairs around 0.34 nanometer, and the whole diameter of this DNA is 2.0 nanometer. So there is a also sugar phosphate backbone, as we have discussed, and when there is making a small turn from here, that's your minor groove, but when it's making a large turn, that's known as the major groove. So and the DNA chain, as we have discussed, it's written there that it is of 2.2 nanometers, and the one nucleotide unit is around 3.3 or 3.4 nanometer of long. So what you are seeing here, like very basics, that these hydrogen bonds are being made between guanine and cytosine. This DNA, the sugar group at the second carbon atom doesn't have hydroxyl group in the DNA, and in the RNA it's present. And we have discussed various nitrogenous base pairs and how in the RNA instead of thymine uracil is present, and we have discussed about nucleosides and nucleotides here, and discussed this antiparallel structure, all about DNA and polymers. So these all things doesn't come in a single day, though it seems these 3 or 4 slides seems very easy to understand. Okay, this is how DNA is made, this is how done, okay, okay. But remember, during the research, when Watson and Crick, they went for the research in their 50s and 60s, it took the ample amount of time to do the X-ray crystallography to find each part, that how things are really is happening, what is really making things to work out. So just my reason here today is not to just clear your basics things, that these things are happening in this way and these things are happening in that way, because this you could also do in your books, or you can go for any Coursera course. There are many courses available in the market. But my reason to be your trainer here today is to analyze, to go in the past in 1950s and 60s, and think from those scientist ideas, from their point of view, just get into into their shoes, and just think how they might be thinking at that time, what difficulties might they have seen in order to come to this conclusion that it's a double helix structure, and these nitrogen base pairs are being centered here. There might be some movie available on YouTube somewhere. I don't know about this discovery of DNA. It will be very interesting to see that how they came out to do such a, because it's a very complex structure. Believe me, if you try to understand, it's a quite complex structure, but they have discovered it, they have made to the public, it's available to the world. That's the DNA is made, and because of that, at the moment in 2021, we can really, believe me, with this COVID virus that's came, what we are doing is we're trying to identify the spike structures and able to identify so that DNA could not mutate in our body. So we are creating so that our immune system could work, and we can create some lymphocytes against them. We're getting these vaccines. So these all things might be not possible because of the molecular biology, and Watson Crick doesn't have found the DNA. So all things make sense at end of the day. So that was some philosophy here. Now comes to the facts. That is ribonucleic acid, that is your RNA. We have discussed basics about DNA so far. Now comes your RNA. So RNA, it's also quite important, very important part, long chain of nucleotide units. It's also have nitrogenous base pairs, ribose sugars, and phosphate. But it's not, might not be as like double helix structures forever. It will be single membrane structure, and then it might be double helix for some time, then again single, and then like this. So at the backbone will be sugar phosphate backbone like DNA, and the sugar will be the ribose sugar in here, and the nitrogenous base pairs will be attached like this here. So there is also double-stranded RNA presence, which is called as dsRNA, which is complementary strands and similar to DNA that found in all cells. So they're mostly present in the viruses. So these we can see some protein codes, these spike proteins. This is the RNA, genetic material inside of it, and present in the form of double-stranded RNA here. So there are some types of RNA. If we see from the perspective of a normal in general, it could be messenger RNA, ribosomal RNA, or transfer RNA. Yeah. Then comes the short forms of mRNA, ribosomal RNA, and transfer RNA, and then there are codes for proteins. And ribosomal RNA needs for the translation, and the transfer RNA also needs for the translation. So currently the Covishield that we are having in India, the vaccine, it's basically working on it has attenuated virus. So virus has been killed, it's not in active form, and it's when it's given into our body, then the lymphocytes, the immune system, the T cells and B cells, they start to work out, and whenever the virus will attack us, so it will help, our immune system will help to fight it. So this one is around 81%, they say, efficient in India that we are getting. The formula is given by the AstraZeneca from the Oxford, but it's being produced in India by Bharat Serum. And the same, but in USA, they are having Pfizer, Biotechnic. They have this mRNA based vaccine actually. So these vaccines has a direct play role, and they are, if this Covishield is around, the effectiveness is around 81%, but this mRNA vaccine based on mRNA based structure, it has 91% effectiveness, even 10% more. So that has been recently published data coming out. So never mind, we still having this 81% effectiveness things into our world. Though there are some controversies saying that the second mutant that we are having in India only, and that what we see the debris of bodies at the moment, um, it's it might be that this vaccine that we are getting, it might be not effective against this virus, or because we don't have any data about that, we don't have anything. So we will see in upcoming 1 month, 2 month, whosoever got the vaccine, so what were the, is there any side effects or something. So we will see how things work. So that's came to me from mRNA vaccine at the moment. And one more thing, I recently went through papers. There's some funny papers are also coming, that instead of going in the AC car, Uber or Ola, go in auto rickshaw. That will be our futuristic mode of transport. Why? Because of ventilation. It's all open. It's all everything is open, and the chances of getting the COVID virus will be much less as compared to the closed car. So they are saying that because of this COVID situation, the main transport of system will be the auto rickshaws in upcoming times. This paper has, and this about this research has been done actually in India by some foreign researchers, that in order to survive for the transport, the ventilation will be the main key for our new world. So this is a new world order that we're gonna live at the moment, where mainly the one who is stronger, who has good immunity, will be able to survive. That's also one conspiracy theories they are saying, and yeah, only the people who have survived will be used from specific task in upcoming time. So let's see what's the task, what's the government is thinking, what's the politics is going behind that. Okay, so that was something about the current situations with the COVID part. Beyond that, these RNAs could also be modified into the post-transcription level, such as small nuclear RNA. Okay, so why RNA? Telomerase RNA. So these are the various RNA presents on the chromosomes, mainly used for the telomere synthesis. On your chromosome, there's a satellite present on the top for that, and the RNA is need for the RNA processing, DNA replications, mostly used in the animals, where the snRNA is used for the splicing and other functions. Splicing, that is to remove your exons and to keep only to remove introns and to keep mainly exons part of your genetic material. And there is also antisense RNA with the transcriptional attenuations, mRNA degradations, stabilizations, and translations blocks. Then messenger RNA. So first part of your types of RNA here is messenger RNA. As name suggests, messenger. So it's act like your the one who transfer one your information from one place to another. So this is a information about the protein sequence to the ribosome. So they take whatever the DNA sequence it says, so it's forward, it's code them into the three nucleotide codes corresponding to the amino acids, and they transfer it to further to your ribosomes and tRNA in order to produce your protein structure. So here we can see we have DNA and we have our messenger RNA, and these are various coding strands on top. So this is, first of all, we will unwind with the helicase enzyme your double helix structure, and then messenger RNA will and it will start to produce corresponding according to the template various parts of your DNA. So this is the non-coding strand, and this will be the messenger RNA with various nucleotides. Then messenger RNA is then exported to the nucleus, from the nucleus to the cytoplasm, and then it will bond to the ribosomes and then translate into the corresponding proteins from with the help of tRNA. So this line that what we it's written, even though I spoke here in just not even, let's say, 10 seconds, but in this line our whole mystery behind your molecular biology is playing a big role here. So what this line says, the mRNA, after getting being transformed into, getting the message from the DNA, then it is transferred from the export to the cytoplasm from nucleus, where it will bind to the ribosomes and then translate it into the corresponding proteins with the help of your transfer RNA. So in order to explain this sentence, let us go to some interesting videos. Transcription and translation. So this is a website from Utah. It's a state in USA, from the university. So they have really cool, very interesting videos, and makes to things understand on a very basic level, to make things clear for us in a very basic way. So let me zoom this.
Transcription and translation are the processes that all living things use to read the information in DNA and build proteins. Each gene in a cell's DNA codes for a specific protein. During transcription, cells make a copy of a gene out of mRNA. During translation, they use that mRNA copy as a blueprint to build proteins out of amino acids. These proteins perform the tasks that make living things function. The flow of genetic information from DNA to mRNA to protein is central to life.
So we have discussed about now the central dogma, that how from DNA to mRNA to protein things are being produced. So let's start. So I have to choose one gene. Let's choose this gene.
Inside the nucleus, RNA polymerase attaches to a gene. It separates the two complementary DNA strands, exposing the gene's unique sequence of bases.
So as we have discussed, within the nucleus, from your DNA, messenger RNA will be produced, complementary. So remember, while doing so, DNA will pair, A in DNA will pair with U instead of T here. So if it is T on the DNA, it will be A. Yeah. What will it will be, students? Tell me in the chatting box. T will pair with A. C will pair with U. A, C, T. T is not there. Maybe G. C will pair with maybe G. T will pair with U, right? U it will be. T will pair with the U, isn't it? Oh no, it's A actually. T with A, and the G will pair with, so G might pair with C. Yes. C will pair with G. So like this, it will continue.
When the end of the gene is reached, transcription is complete. The new strand of mRNA passes out of the nucleus and into the cytoplasm, where translation takes place. The instructions in an mRNA strand are read in groups of 3 bases called codons. Each codon must be translated, one after another, in order to build a functioning new protein. The key structures in that process are the ribosome and transfer RNA, tRNA. Each tRNA molecule has its own anticodon and a matching amino acid, the building blocks of proteins. To begin translation, a tRNA molecule coded for the amino acid methionine docks with a ribosome. The ribosome attaches to the end of the mRNA strand. It slides along until it finds a start codon, AUG. Through complementary base pairing, the start codon pairs with the anticodon on the tRNA molecule, signaling the beginning of the protein instructions. The methionine molecule is now the first amino acid in the new protein. The ribosome shifts to read the next codon. Each codon is recognized by one of 20 different tRNA molecules. Each type of tRNA carries a different amino acid. The ribosome continues moving along the mRNA strand, one codon at a time, matching it to a complementary tRNA. It adds each amino acid to the growing protein in just the right order. The sequence of amino acids determines the final shape of the protein and the function that it will carry out. Give translation a try.
So, participants, we have discussed your mRNA production just now from the DNA, where A will correspond to U and T will correspond to A. Yeah. Now we will discuss further that from this mRNA has been exported from your nucleus to cytoplasm, like the last sentence that we have seen, and that being transferred to the cytoplasm with the help of your ribosomes and tRNA, and it will correspond to specific codon, anticodon, and will produce various amino acids in the form of 3 base pairs here. So here we can see that the first, it starts with the methionine initially, that's a start codon, and then at the end it stops by the stop codon. So here if we have GUU, so what should be the next tRNA that we should use against this mRNA? Whether it will be leucine, valine, or another form of valine, or another form of valine, which might be the possible transfer RNA do you think we should take here? So it's G is here, then it's correspond to maybe C, and then U is here, then it might be correspond to A, then C, A. So it seems this one quite fine. Yeah. So because of that, our valine amino acids being there in the ribosome on the top, And the same way, so with the help of AUG, is what is happening is initiation. And now after the initiation, elongation step will be there. We will keep adding your further things. So here we have now CGU. Yeah, we have our CGU. So C will be G, then C, and then A. So it might be arginine. Yes. So valine was there, arginine is there. Now comes CUG. So this will be, students, which one it will be? Tell me. CUG. CUG. Tell me whether it will be CUC, GCU, CCC. Will it be glutamic acid, leucine, arginine, glycine? Yeah, it will be leucine. So in this way, our, you can see this, there's a small polypeptide chain of amino acids being made here very nicely. So let's make it to forward.
When the ribosome reaches a stop codon, it detaches from the mRNA strand. The chain of amino acids folds into its final form and the new protein is complete. This protein, aminoacyl tRNA synthetase, will now attach the right amino acid to the proper tRNA molecule, charging the tRNA so that it can participate in protein synthesis. Each type of amino acid has its own enzyme. The incredible processes of transcription and translation enable cells to carry out all of the functions necessary for life.
So I hope this small video might have explained forever in your life, that you will never forget that how transcription and translation works. So I will share this link with you over the what? There's so many things here. I sometimes forgot. Oh, there's a Delhi West. Now just pin this for future. So yeah, oh, I should have seen some questions. You might be having some questions there. Oh no. This chat is going very slow. Sir, please can you explain histone proteins and its octet orally? Yeah, Ghanshyam Tiwari, very good question that you have proposed here. Um, because we are in the flow of molecular biology at the moment with the RNA and transcription and translation. Once we are done with this topic, please do make me remember to explain how your DNA is coiled around the histones. I have one presentation with me, so I will share that with all of you here. So yeah, so my point is being clear here now. What I want to say that that how your messenger RNA and DNA playing a big role in the central dogma in order to produce your protein, the polypeptide chains of amino acids. Whereas in the prokaryotic cells, which do not have nucleus and cytoplasms compartments, so mRNA can bind to ribosomes where it will be transferred from the DNA. So it's a different concept in the prokaryotes. Then transfer RNA, after messenger RNA comes your transfer RNA, that is which is a smaller chain of about 80 nucleotides that transfer specific amino acids to growing polypeptide chain at the ribosomal site of protein synthesis. So we have discussed how this tRNA attaching to the mRNA, having these anticodones at the below, and one specific amino acids being attached on the top, and working with playing role with the help of your ribosome. Then comes your third type of messenger RNA, that is your RNA, that is your ribosomal RNA, which is quite a catalytic component of your ribosomes, and it is of majorly two types, that is 70S and 80S, made up of respectively 50 and 30, 60 and 40S. So in the cytoplasm, ribosomal RNA protein combine to form a nucleoprotein called ribosome, and ribosomal RNA is extremely abundant. Is extremely abundant. Yeah, makes around 80% of your 10 milligram per ml RNA found in the typical eukaryotic cytoplasms. So difference between DNA and RNA, that is RNA is your nucleotides that contains ribose sugar, whereas the DNA contains deoxyribose sugar. And RNA has a base uracil, where DNA has a base thymine. And on the other side, RNA has a presence of hydroxyl group at the second position of the ribose sugar, whereas the DNA lacks the hydroxyl group of the second position of the ribose sugar. And RNA is usually single stranded and DNA is your double stranded. So I will stop here from the today's point of view, the lecture. And there was a question that somebody asked to explain about histone, how the histones are being carried out over the DNA. So it was in one of my presentations, cytogenetics, I was teaching to the college. Just a second. No. Yeah. So somebody was asking me about how your DNA is all being compacted and how everything is done in that part. So here we can see that first of all, in the chromatin structure, your DNA is being there, and they're coiled around your histones, and this we have discussed, and it's become a nucleosomes, and they become 10 nanometer beads on a string fiber, and they get further get folded. Scaffold proteins are made with 30 nanometer fibers, and then further, and then they are being at the chromosomes, like beads on a string. It's also called as a beads on a string. So my point is here to tell you that how the histone is being made actually. So basically your histone are the major components of a chromatin, and there are four families of histones that are present, that is your H2A, H2B, H3 and H4. Remember them: H2A, H2B, H3 and H4. So an additional non-core histone um, is also there that we have discussed on that acts like a clip to hold everything together, that is your H1. So what happened basically, there is a H3 and H4, they make a dimer, and then H2 and H2B, they make a dimer. Yeah, then this H3 and H4, they make a tetramer, like these together, they join together to make a tetramer, and this H3 H4 tetramer plus H2A and H2B dimer, they come together to make a proper histone octamer. And around this histone octamer, your DNA is being coiled like this, complete histone with DNA. So that's the basic thing behind your yeah, chromatin structure or histone structure. So here you can see this is a histone octamer with H2A in the center, H2OB on the left, then H3, H4, H3 and H4. So like this, your whole octamer structure being shown like this. And this is the top view from it, and this is the side view of the same. So I think, and this one, that's I want to show you the H1 histone. Here you have an octamer of histones here, 8 histone octamer, and then and your H1 histone is on the side to let the entry of your DNA and the exit of your DNA. The C tail of H1 stabilize the DNA between the nucleosome cores here. It's helping the stabilization of it mainly. So I think this is more hardcore things. I think we should not go further. So I will make the next topic, that is your protein, or maybe you could take your questions now. Shruti Mandal, as I mentioned, what you mentioned. Can we accept a genetic code with 2 letters rather than 3 letters? I somewhere read that it leads to less protein diversity. Oh, that's a new field of study. This might be the mutation actually. If we have not 3 codons, so they will not code for protein, your proper genetic code, the protein that we are working on. So this might be not good enough to do the functions that they should do. So I need to look for that, but that will be a genetic mutations if we have um, 2 letters of your genetic code. Okay, I hope Ghanshyam Tiwari, your question about histone proteins and octet, how it works, might be clear in that those slides with H2A and H2B coming together, and H3 and H4 they're coming in a tetramer, and they're coming together, they're making an octamer, and the H1 will be on the right hand side, which will be acting as a to make the stability of a protein so that the DNA enters and DNA exit from it. So that's how the whole histones being played role here, and this whole system of DNA is known as a bead on a string. Yeah, it's known as a bead on a string. So can we accept a genetic code with 2 letters? So this is done. How, but actually how is codon with 2 letters even possible? Yeah, that's mystery. Mhm. So Siddhi Trivedi, what topic do you think like I should explain? Which, please let me know which things you, otherwise you can write me later. I can. Oh yeah, that's Nidhi Kulkarni. Yeah. And there are always, exceptions, like in the Mendel thing that we have studied. Um, there were later on some exceptions like codominance. Um, many things came out later. So the same way in the Wobble hypothesis that proposed that normal base pairings can occur between nitrogen's base pair in the positions of 1 and 2 of the codon. Yeah, and the corresponding bases in the anticodons. So in that case, the one anticodon can form non-Watson-Crick base pairing with the third position of codon. So that's true. This two base pairing thing in the Wobble hypothesis might be, yeah, create some problem actually for the tRNA, because Francis Crick actually he proposed this vowel hypothesis, even though they discovered that mainly three base pairing is being done. That's true, but he postulated that the 5 base pairing on the anticodones, which binds to the 3' base pair of the mRNA, could be separately confined to the other 2 base pairs, leading to have non-standard base pairing. So that's also possible. Yeah. Okay. So we are done for today. That's it. Not too into details. So we have just discovered. I will summarize that we have just talked about mRNA, various types of your sorry, RNA, mRNA, tRNA, ribosomal RNA, and we have discussed in that video how your transcription and translation is being playing a role. And beyond that, about we have discussed about yours histone formation, how the histones are playing a role. Um, yeah, and we have discussed today about DNA, how the various structures, how everything is pairing and making sense at the end. So that's the very basic thing that we have discussed today. So we will discuss from tomorrow how your proteins replications, DNA replication is being playing role in that. So I see some question by Shruti. Want to keep your ink continuations? Discard? No. About translation and transcription. Okay. You can message me later. We can have a chat over it, or some notes I can provide. But at the moment, uh, some Shruti gave the notes. Very nice. DNA double helix around histone proteins H2A, H2B, H3, H4. So it's a 3/4 round of DNA. Very nice. This is how they are arranged. You can see in this scare box that we have seen in that diagram. Very nice, Bache. I like your diagram a lot. It's basically explained that you have understood the concept. The person who could understood the concept only could draw these things um, like this. I'm liking this diagram a lot. So this H1 clips DNA to histones. That's true. This H1 on the side clips to it. This is the whole nucleosomes, histones per DNA. Very nice. The nucleosome plus DNA, linker DNA plus H1, it makes your chromosome. So somebody was asking yesterday that what is the difference between chromosome and chromosome. So that's the difference, bache. Then these becomes bead-like structures of chromosomes, nucleosomes. So they become started to go further, further, further. And this is oh, I will save this for myself actually in the future teaching. Then it's become histone protein polymers, and then you can see these wobbling and getting all these being structural, the spring loop like structures, they call solenoid structures of the nucleosomes. And then they got scaffold, they get into the nucleus, and then at then this is a scaffold proteins. But where's the chromosomes? Yeah, then it's become a chromosome and then centrosomes. So this is third coiling, this is the second coiling, and this is the first coiling. Super nice. Thank you, Shruti, for sharing this thing. So I hope some students could also learn from it. Yeah, lack of operon. We can discuss about that in details. There's a special lectures in genetics about that. Lack of operon, TRP operon, regulatory factors. It's an interesting thing. It's a different topic. We can discuss about that on some other day. Yeah, yeah. Okay, so that's it for today. Yeah, you might have another classes now, and then we will meet tomorrow, same time, same place. Or maybe not. Tomorrow is Friday. Maybe there will be no class tomorrow for some personal reasons. but I will compensate that class into the next week Saturday, so that we make things done. So we might see from now onwards, on Monday itself. And if you have any questions, anything that want related to review article, there was some on my Telegram at 1:30. I got some message from a student, but I had to make a lot of question papers today for the online exam that is coming up, so couldn't entertain any students today. It was very difficult. But during this tomorrow time, I can you can call me anytime, and I will be happy to answer your review questions, articles, how to write a review article, how to write nice one. So I can help you in that. So tomorrow there will be no class. I will compensate that class into the next day somewhere. Okay, great. Thank you very much. Stay safe, stay healthy. Uh, Take care then. Yeah. Bye bye.